Hello, dear student. I am your educator Shrutika, and today we are going to discuss the squash preparation of the onion root tip for mitotic stages. So, let's start the topic. So, aim of the this practical is to understand the process and different stages of the mitosis and to visualize different phases of the mitosis. So, in this we are going to study the different stages of the mitosis. Then moving to the principle, the genetic information of all organisms reside in the individual DNA molecule. You all know that. Okay. So an onion cell possesses 8 chromosomes, and whereas the human cell possesses 46 chromosomes, 23 pairs. So in 1842, C. Nageli first saw chromosomes, and in 1888 W. Aldair named them, and then the Walther Fleming studied and named the process of a cell division as a mitosis. So cell division occurs rapidly in the growing root tips, sprouting seeds or bulbs. Do you understand? Do you understood this sentence? That the cell division occurs rapidly in the growing root tips and sprouting seeds or bulbs. Means the cell division, we can see the cell division with the help of the slides, mitotic slides, mostly in the growing root tips of sprouting seeds or bulbs. So for example, I am taking here a onion, that is Allium cepa, or let's say the garlic, that is the Allium sativum. So I have taken this Allium sativum, and here this is the Allium cepa. So in case of Allium cepa or in case of the Allium sativum, what you see, when you place these plant specimen, when you place these bulbs of a Allium cepa or sativum on a glass beaker or anything, if you are growing them in your home, then if you place them in a container for sprouting, then what you will notice that from this region there will be numerous growth of a root tips, and when you cut these root tips and watch it under the microscope, then you can visualize the process of the mitosis or the process of the cell division. So an onion root tip is rapidly growing part of the onion, and thus many cells will be in different stages of mitosis. Okay, and the onion root tips can be prepared and squashed in a way that allows them to be flattened on a microscopic slide, so that the chromosomes of the individual cells can be observed easily. The super coiled chromosomes here during the different stages of mitosis present in the onion root tip cells can be visualized by treating with the DNA. Um, by treating with DNA specific stains like Fulgen stain and acetocarmine stain. Okay, so now let's see what are the materials required for this practical. Firstly, the onion plant with root, obviously. Then any stain like acetocarmine. Then one normal HCl, scissors, forceps, razor blade, microscopic slides and cover slips, and water bath. And you will need a light microscope. Now let's discuss the process. So firstly you have to cut the tip 5 to 8 mm from the tip of a freshly sprouted root. So for example, this is a root of what? Onion. So this is onion for example, and this is sorry, this is onion, and this is root of onion. So it can be best studied when the roots are not very much grown or not very much short. Then how much should be its size? So it should be 5 to 8 mm from the tip. Okay, it shouldn't be too much like that. So it should be just of a proper length, and from this you have to cut only 5 to 8 mm. So 5 to 8 mm from the tip of the freshly sprouted root. Then you have to discard the remaining root, and later you have to wash it in a water or a clean microscope slide on a on a clean microscope slide. So when you take a microscopic slide and you put this root here, then you have to cut it from the, this is a tip part, so you have to take this tip part. So after cutting it with a blade, you are cutting it with a sharp blade. Later let's draw the slide first. So here you have discarded this part and you have taken only the root part. Okay, now you have to wash it with a clean wash it with the water. Okay, and after that you have to place one drop of 1 normal HCl on the root tip. So pour 1 normal HCl on the root tip, then add 2 to 3 drops of the stain. So you have to add 2 to 3 drops of acetocarmine. Then you have to warm this slide gently on the alcohol lamp for about 1 minute. Okay, then carefully you have to blot the excess stain with the blotting paper. So here in the protocol, one thing should be added that I think it is missing in this protocol here. So okay, so after that, before warming the slide, what do you have to do? You have to add cover slip here. So before which you have placed 1 drop of 1 normal HCl on the root tip, then 2 to 3 drops of acetocarmine stain, and after that you have to add cover slips and warm the slide gently over the alcohol lamp. After that you have to carefully blot the excess stain with the blotting paper, and after 10 to 20 seconds put 1 or 2 drops of water and blot them carefully using blotting paper. Then again you have to put a drop of water on the root tip and mount a cover slip on it, avoiding air bubbles. I think here there is some mistake here in the protocol. You have to squash it also. Acha. So okay, now let's read it. So squash the slide with your thumb using a firm and even pressure. So what is squashing? So when you put a root here, then cover slip here, then you have to squash it with a thumb or your needle. So this is, for example, a needle. This is the pointed part and this is the blunt part of a needle, and you have to squash it with that needle so that it will the chromosome will be separated easily. And and one more thing, you don't have to break this cover slip and distribute all the chromosomes. With a gentle pressure, after heating, the chromosome will be separated by their own. So don't apply too much pressure here, neither the cover slip will be broken. Then observe it under a microscope, compound microscope, in a 10x objective lens, and scan and narrow down to a region containing dividing cells and switch to 40x for a better view. So the process of mitosis is divided into 4 stages: prophase, metaphase, anaphase and telophase. Okay, as you all know. Now let's see the different types of phases in detail. So firstly, the prophase. So during this stage, the chromosomes supercoil, okay, and condense and become visible for first time during the cell cycle. So this is the cycle where you can see condensed chromosomes. These spindle fibers start forming here, okay, and the nuclear membrane starts disintegrating. So this is the nuclear membrane. It starts to disintegrate and there is formation of the spindle fiber. So for what purpose? Yeah, because this cell is going to divide. What that means? This cell is going to divide, that means the chromosome will divide themselves and they form daughter chromosomes, so that when our cell division will be over, then the chromosome will be likewise they will be separated. So firstly the prophase. Okay, so we have discussed the prophase. What happened in the prophase? Formation of the spindle fibers and disintegration of the nuclear membrane. And how can we identify this prophase? Because the chromosome present here are in the supercoil form, okay. And then now moving to the second phase of the mitosis, that is the metaphase. So during this stage the spindle fibers reach and attach to a centromere of each sister chromatids, and the chromosomes align along the center plane of the cell, and the nuclear membrane disintegrates completely. So completely here only started disintegrating, and here it is completely disintegrates. So you can see here also. Okay, we will discuss it later. Now the anaphase. So during this stage the centromere starts splitting and the sister chromatids begin to migrating towards the opposite poles of the cell, okay. So what happens here? the centromeres start splitting. See, okay. Suppose this is a chromosome. When this is a spindle fiber, and here the centromere, so it is attached to centromere here, and this is here. So what happens, this will move here on this pole and it will move towards this pole, and likewise the sister chromatids begin to migrate towards the opposite poles of the cell. Then the last one, that is the telophase. So here in the telophase, during this stage the chromosomes are clustered on either end of the cell, and the nuclear membrane starts reforming. So here the nuclear membrane, which was completely disintegrated where in the metaphase, and started from prophase. So here the the nuclear membrane starts reforming, and the chromosomes are clustered here on either end. So here the chromosome will be in the clustered format. Likewise. And this new cell wall, okay, starts to form between the two daughter nuclei. So in case of plants, what happen, okay, that is different parts, we will discuss it later, okay. So now let's discuss this diagram. So in case of interphase, what is interphase? When the cells are in the non-dividing stage, or we can say the G0 stage. I think let's take this color. So here the cells are in the G0 stage, non-dividing condition. So in the early prophase, so what happened in the early prophase, you can see the chromosome as in a condensed form in case of early prophase, and in case of mid prophase, you can see chromosome start to dissociate, or we can say start to being loosely packed. Here again also they become loosely packed. Then in case of metaphase, they started to they come at the equatorial plane, and here in the prophase there is a disintegration of this nuclear envelope. So what is this? This is a nuclear envelope, and here it disintegrates. This is complete formation of what? Spindle fibers, and these chromosomes come in the center in case of metaphase. So you can identify metaphase. Likewise, this is the early anaphase. These chromosomes start to separate, and in case of late anaphase, you can see they are present just near to the two poles, and in case of telophase, they completely present at the poles, And in case of late telopes, they form a nuclear envelope around themselves, and um, there is cytokinosis, that is division of the cytoplasm also. And up to this is a nuclear division, and from this you can see there is a starting of the cytokinosis by forming a cell plate. So this is a cell plate. Okay, I think here we should end. One more thing, that this will form two daughter cells from a single cell nucleus. So this is the squash preparation of onion root tip for mitotic stages. One more thing I would like to add here, that some plants like let's take ginger or Costus speciosus. Okay, what is Costus speciosus? It have also tubers or rhizome like the ginger. Okay, so for example, this is the tuber or rhizome of the Costus speciosus. Costus speciosus is a beautiful plants having the beautiful flowers. So in case of Costus speciosus, this is its rhizome. When we place it in a water. Okay, so why we are placing it in a water? Because it will not germinate. If we place it in a sand or let's say clay, etc., it will not germinate easily. I have done its practical, but it doesn't have um, germinated properly in that environment. And when I put it in a water vessel, it started to germinate by just 3 to 4 days. So what I was telling you, that plants like these, ओनियन का तो सवाल ही नहीं. ओनियन में सिंपल यू हैव टू कट द roots, but in some plants like that, what we will see, you will notice that it have roots as well as root hairs. So you don't have to cut the root hairs, you have to cut the roots. For what? This squash preparation. Because, or we can say the cytology purpose, because when you cut the root hair, you will not see anything, and when you take proper cutting of the root hairs, the roots, then you will see good chromosomal segregation, or we can say the cell division, for which purpose you are doing the practical. Okay, so this is all about the squash preparation of the onion root tip for the mitotic stages. So here we have completed this practical. Hope you have understood the lecture. Please don't forget to like, share and subscribe the channel. Thank you.